We'll just start in a few minutes. We'll wait for if anyone else joins and then start in a few minutes. Okay, so I'll share my slides. Meanwhile, if anyone wants to join, please admit them. Okay, so welcome everyone to the third live TA session. So in this session, I will be discussing about the topics that were taught in week 3, which was basically the introduction to structural biology techniques and X-ray crystallography. So firstly, I'll discuss a summary of what Sir had taught, and then we'll solve some sample questions. So feel free to stop me at any time with your doubts. Okay. So first, we will learn about the hierarchy of life. So all living and non-living things are made up of one or more unique substances called elements, the smallest unit of which is the atom. Atoms combine to form molecules, which are the building blocks to all structures in the human body. Now, as you know, from the molecule, many molecules can come together and form a macromolecule. And in the previous class, we had learned that there are 4 kinds of macromolecules, which are carbohydrates, fats, proteins, and the nucleic acids. So from the molecule, you get the macromolecule. Now, many macromolecules come together to form an organelle. And many organelles come together to form cell. So all living structures are made up of these cells, which are made up of many different molecules. Cells are the smallest independent living thing in the human body, and the body is made up of many different cell types, each with a particular function. In multicellular organisms, these cells can be organized into tissues. A tissue is basically a group of similar cells that work together to form a specific function. Then from the tissue, the next level is the organ. And an organ is an identifiable structure of the body composed of 2 or more tissue types. Organs often perform a specific physiological function, like, say, digestion or excretion. An organ system is the next level of organization, and it is a group of organs that work together to perform a specific function. And the final and most complex level of organization is the human organism, which is composed of many organ systems that work together to perform the functions of an independent individual. Next, we will talk about the two-component system. So these are basically found in prokaryotes, and in molecular biology, the two-component system serves as a basic stimulus-response coupling mechanism, allowing the organisms to sense and respond to changes in many different environmental conditions. They typically consist of a membrane-bound histidine kinase, shown here, that senses a specific environmental stimulus, and a corresponding response regulator that mediates the cellular response. So how does this signal transduction occur? So the signal transduction occurs through the transfer of a phosphoryl group from adenosine triphosphate or ATP to a specific histidine residue shown by H here in the histidine kinase. So ATP becomes ADP and transfers the phosphoryl group to the histidine. This is called an autophosphorylation reaction. Subsequently, the histidine kinase catalyzes the transfer of this phosphate group on the phosphorylated histidine to an aspartic acid residue on the response regulator. The phosphorylation causes the response regulator's conformation to change, usually activated an attached output domain, which then leads to the stimulation or repression of the expression of target genes. Such two-component signal transduction systems enable bacteria to sense, respond, and adapt to a wide range of environments, stressors, and growth conditions. So the two-component system that you learned is basically how a system communicates within itself from the stimulus to the response. But we need to understand the atomic level of this communication in biological systems. So in biology, communication is mostly performed through interactions. And in order to understand the process of this communication, we need to visualize the molecules in the atomic level and determine the proper arrangement of the atoms in these systems. However, as we have seen before, there are several different levels of interactions, as well as the size of macromolecules or their organizations, which are quite different. So biological systems possess complexity, as you saw in the hierarchy of life. So we have to identify the layers so that our understanding can be made at the fundamental level. So to study this communication at the fundamental level, we need a high resolution picture so that we can understand the mechanism at the atomic level. This high resolution picture would give us an understanding of the mechanism of action of macromolecules at atomic level. And this understanding is crucial to understand how diseases happen and to develop new drugs and engineered systems. So there are several high resolution techniques to solve the structure of biological macromolecules. In the 20th century, a variety of experimental techniques were developed to examine the three-dimensional structures of biological molecules. The most prominent techniques are X-ray crystallography, NMR, which is nuclear magnetic resonance, and cryo-electron microscopy. So as you can see here, the sizes of several objects are shown, from small molecules to cells and eukaryotic cells. And the range of different spectroscopic methods are shown. So NMR spectroscopy is ideal for detecting small molecules. X-ray crystallography can extend a bit and detect up to proteins and proteins complexes. Single particle cryo-electron microscopy and cryo-electron tomography can detect anything from proteins up to cells. But as you can see, the resolution of light microscope is not enough to study macromolecules. That is why the discovery of X-ray was important, and its applications to protein crystals revolutionized structural biology, as scientists could now obtain the three-dimensional structures of biological molecules in atomic details. Likewise, NMR spectroscopy also allowed information about protein structure and dynamics. And finally, in the 21st century, electron microscopy also saw a drastic revolution with the development of more coherent electron sources aberration correction for electron microscopes, and software that reconstructed the pictures and enabled successful implementation of high-resolution cryo-electron microscope. This permitted the study of individual proteins and molecular complexes in 3 dimensions up to angstrom resolution. So if we look at the 3 methods, X-ray crystallography, NMR, and cryo-electron microscopy, these are some differences between the methods. So in X-ray crystallography, the sample must be crystallized in a lattice structure. But in NMR, the sample can be in solution or solid state or membrane bound. So it has a wider range. In cryo-electron microscopy, the sample must be frozen in its native state. Now, X-ray crystallography can detect any size molecules. But NMR can only detect small molecules which have less than 25 kilodalton weights, while cryo-electron microscopy is used for larger molecules. The resolution of X-ray crystallography is near that of atoms, which is 1 to 3 angstrom. NMR has slightly higher resolution, which is 2 to 3 angstrom, while cryo-electron microscopy has even higher, which is 3 to 4 angstrom. NMR, solid state NMR, can be used to solve membrane proteins. And cryo-electron microscopy, the sample preparation is very fast, which is the advantage of cryo-electron microscopy. So these are the 3 main techniques to solve the structures of biological macromolecules. Now, in general, in all the techniques, there is something called a phase problem. To understand that, we first need to understand that what is the amplitude and what is the phase. So the amplitude is the distance from the rest position to the peak or the crest position, which is basically half of this vertical distance from the trough to the crest. So this is the amplitude. And phase involves the relationship between 2 or more signals that share the same frequency. So phase involves the relationship between positions of the amplitude crests and troughs of 2 waveforms. So here you can see 2 signals, and the phase difference between these 2 signals is 90 degrees. So it is said that these 2 waves are 90 degrees out of phase. So what is the phase problem? So from diffraction experiments, you can get a pattern like this, which is basically you are getting the intensities. And from intensity, you can get amplitude because intensity is amplitude squared. But all the information about the phase is lost. So that is a problem because if you don't know the phase, you cannot perfectly reconstruct the atomic positions. So that is what is the phase problem, that from the diffraction pattern, you do not know the phase. And the knowledge of both the amplitude and phase is absolutely essential to reconstitute the electron density of the crystal that gave rise to this diffraction pattern. So how is this phase problem solved? We'll deal with that later, but this is just the introduction to the phase problem. So what is the goal of X-ray crystallography? Basically, in order for an object to be visible under magnification, the wavelength of the light must be, roughly speaking, no larger than the object. So visible light, which is in the range 400 to 700 nanometer, cannot produce an image for protein molecules in which the bonded atoms are about 1.5 angstrom or 0.15 nanometer apart. So as you can see, the size of the protein molecules is here, and the wavelength of the visible light is greater than that. So visible light cannot be used to study macromolecules like proteins. However, the X-ray wavelength fall within the range of the proteins and atoms. That is why X-ray crystallography is needed to visualize these macromolecules. So there are some associated problems with X-ray crystallography. The first of which is why we can't visualize the molecule directly using X-ray. Why at all do we need the crystal? So a single molecule is a very weak scatterer of X-ray. So most of the X-ray will pass through the molecules without being diffracted, and the diffracted ray is too weak to be detected. So from a single molecule, if we do the X-ray, then the intensity will be so low that we cannot reconstruct the atomic positions. That is why from the molecule we get the crystal, because a crystal is made up of three-dimensional repeats of ordered molecules, which can be as much as 10 to the power 14 molecules. And so the diffraction from crystals is such that the intensity becomes much more. And so crystals are used and not single molecules for X-ray diffraction. The second point is that unlike visible light, X-ray cannot be focused by lenses. So what is the principle that allows us to observe something in a light microscope? That is basically the incident ray is coming through air, suppose, and when it hits the lens, the refractive index of the lens is different. And so the visible light bends. And like this, in the microscope lens, the visible light rays come and they get focused at a certain point called the focal point. And this is the principle of the light microscope. Now, the problem with X-rays is that the refractive index of X-rays in all materials is very close to 1. What that means is the speed of the light, the speed of the X-ray in air and in any material is almost the same. So the X-ray will not bend when it passes through the lens. So if the X-ray does not bend, it cannot be focused. And that is a big problem, that so we cannot directly use lens to focus X-rays and get the image, because X-rays will not bend and be focused at the focal point. So what is done to solve this is basically a computer is taken to simulate an image reconstructing lens, and the computer plays the part of the objective lens, computing the image of the object and then displaying it on a screen. Now, how are we sure that the structure that is obtained from NMR and X-ray is similar? So several experiments were done. Firstly, on the phage 434 repressor protein, and this blue color that you can see is the data from X-ray, and the orange color is the data from NMR. As you can see, the two methods produce very similar prediction models for this phage repressor protein. Similarly, in the tendamistat protein, again, this X-ray and NMR structures are very similar. So this is a proof that both these techniques are valid to find the structure of a protein. Now, we need the crystal. As you saw in the last slide, we cannot directly use the protein molecule, but we need to crystallize the protein molecule. So in an ideal crystal, which is a perfect crystal, every atom has identical surroundings. However, real crystals are not perfect. So a real crystal always has a large number of imperfections in the lattice. So the imperfections can be of many types. Like this is a perfect lattice, but it does not exist in reality. In reality, there can be some impurity in the space between the atoms, which is an interstitial impurity. There can be a space where this red atom, say, is absent, or the green atom is absent. So that is called a vacancy defect. Or it can be that instead of this red atom, some other atom has entered. Or instead of the green atom, some other atom has entered that position. So that is called a substitution defect. Or it can be that instead of the green atom, instead of the red atom at this position, one of the green atoms occupies it. So that is called an anti-site defect. So there can be so many different kinds of defects in real crystals. So why are there defects? So basically, real crystals are of a finite size, and so they have a surface to their boundary. At the boundary, the atomic bonds terminate, and hence the surface itself is an imperfection. So you can reduce the crystal defects, but you can never eliminate them entirely in a real crystal. There are certain differences between small molecule crystals and protein crystals. One of the notable differences is that in protein crystals, the solvent content is very high. Typically 30 to 80% by volume of the crystal is solvent. Now, because of this, the protein crystals are very soft and fragile and easy to crush. Because in the crystal, the crystal contacts which stabilize it are very low. Because of the high solvent content, most of the protein surface is in contact with solution. So it is very easy to crush. Another thing in protein crystals is that small conformational changes can take place within the crystal lattice without damaging the crystals. And sometimes even large structural changes can be accommodated. That is why enzyme crystals are often active as catalysts. So if you look at the diagram here, this is an enzyme crystal. And this is the active site of the enzyme. And when the substrate is coming, the active site is able to conformationally change its shape so as to bind to the substrate. So even in the crystal, this flexibility is there, which is allowing the enzyme to change its shape to bind to the substrate and to form the products and regeneration of the enzyme. That is why even in the crystalline form, enzymes are active. Another thing is that in protein crystals, there are certain anisotropic physical properties like birefringence due to the anisotropic refraction indices. So birefringence is basically when you have an incident light and due to the difference in refractive indexes, it is split into two beams of light when it exits the protein crystals. Which in this example, you can see that the incident beam has struck the crystal, and when it emerges from the crystal, there is a horizontally polarized beam as well as a vertically polarized beam. Now in protein crystals, the refractive indexes are different, and so this can happen, but this does not happen in case of small molecule crystals. Next, we will talk about DNA to protein. And so basically, before crystallizing the protein, we need to express it and optimize this expression. So for this, E. coli is used. So why E. coli is used and not some multicellular organism? Firstly, there is no ethical concern about growing, manipulating, and killing the unicellular bacterial cells. But if you used a multicellular organism, there are many ethical concerns. Secondly, most of the strains of E. coli are harmless, so there is no chance of any disease spreading from these experiments. Another thing is that unicellular organisms like E. coli are able to reproduce and grow very rapidly, doubling its population about every 20 minutes. So this is helpful in research to get many generations within a short time. But multicellular organisms generally take a long time to reproduce, so this will slow down the research considerably. Thirdly, E. coli can survive and are adaptive to variable growth conditions and can be manipulated and engineered easily. So these points make E. coli very feasible for the expression system. Next, for cloning vectors, something called plasmid is used, which is basically a circular strand of DNA, which is self-replicating. So why is plasmid used? Firstly, its small size makes it easy to manipulate and isolate. Secondly, it is circular and it is hence more stable than the linear DNA. Thirdly, the replication is independent of the host cell, and so several copies of the plasmids can be present. And fourthly, plasmids frequently have antibody resistance. So when you insert your gene of interest into the plasmid, you can specifically filter out those which have antibody resistance, having your gene of interest from the other plasmids. So things like this antibody resistance is called a selective marker, which enables you to identify the plasmid that you want from the rest of the plasmids. And plasmids also have something called an origin of replication, which is a non-coding DNA sequence that defines the position from which the pDNA replication commences. So this gene of interest that you want is inserted after this promoter region. So how is the plasmid used for DNA cloning? Firstly, what is DNA cloning? DNA cloning is the process of making multiple identical copies of a particular piece of DNA. In a typical DNA cloning procedure, the gene or other DNA fragment of interest is first inserted into a circular piece of DNA called the plasmid. So say this is your target gene, which here is the insulin gene, and this is your plasmid. So they are both digested by restriction enzymes. And what this does is that the sticky end of the plasmid and the target gene is similar. So that you can see this end will join easily to this end, and similarly for this end. Then they are joined by DNA ligase. And this is called recombinant DNA, which is basically DNA assembled out of fragments from multiple sources. Next, this recombinant plasmid is introduced into the bacteria, and the bacteria carrying the plasmid are selected and grown up. So when the bacteria reproduce, they replicate this plasmid and pass it on to their offspring, making copies of this DNA. But and then what you can do is that you will have a colony which has the normal plasmid, which is the cloning vector colony, and it also has recombinant plasmids, which is shown by these white circles here. Now, generally in the recombinant plasmids, a selectable marker like you saw in the previous slide is inserted, which in this case is antibody resistance. So now if you put an antibiotic, the non-recombinant plasmids will die out and you will only have the recombinant plasmids. So you have managed to get a colony of plasmids which have only your gene of interest. But what is the point of making many copies of this DNA sequence in a plasmid? So in some cases, we need lots of the DNA copies to conduct experiments or build new plasmids. In other cases, like this insulin gene here, the piece of DNA encodes a useful protein, and the bacteria can be used as factories to make this protein. For instance, the human insulin gene is expressed in E. coli bacteria to make insulin by diabetes. Now that you know how to make the protein, basically by inserting the gene and then allowing the bacteria to reproduce, and then the subsequent generations of bacteria will naturally synthesize the protein. So now that you have the protein, you need to purify it. So there are several methods of doing that, one of which is gel filtration chromatography. In this, porous gel beads are taken and you add your sample mixture, which has both large molecules and small molecules. So in this porous gel beads, the pore sizes are such that the small molecules can enter the pores and remain inside, while the large molecules will be eluted first. And then you can add extra solvent so that the small molecules are also eluted. So the separation in gel filtration chromatography is done by the size. And as the column flows, the large proteins are excluded from pores and they flow out rapidly, while the small proteins enter the pores and will flow out later. So this is separation based on size. You can also separate based on charge, which is called ion exchange chromatography. In this, the column has certain negative charges which are bound to the column and positive charges which are free to flow. Now, if you add your protein mixture, which has both positively charged proteins, which is basically say basic amino acids, or negatively charged proteins, which is acidic amino acids. So now when you add this protein mixture, the positively charged proteins will be immobilized that is, it will remain on the surface of the column attached to these negative charges. but your negatively charged protein will collect the free positive charges and flow out. Next, if you add certain amount of salt like NaCl, that will neutralize your positively charged proteins, and the positively charged protein will flow out next. So basically in this column, what you are doing is that you are separating based on charges. So the different proteins have different binding affinity, meaning the positively charged proteins is getting stuck to the column, this negatively charged column. So it is being separated from the negatively charged protein. And the positively charged protein comes out later and the negatively charged protein comes out first. So in this way, you are separating the proteins based on charge. Another method is called affinity chromatography. This is based on highly specific biological interactions between two molecules, such as interactions between enzyme and substrate, receptor and ligand, or antibody and antigen. So these interactions, which are typically reversible, are used for purification by placing one of the interacting molecules, referred to as affinity ligand, onto a solid matrix to create a stationary phase, while the target molecule is in the mobile phase. So one example of this is nickel affinity chromatography. In this, the basic the thing is that you have certain beads which have this nickel substituent, and the beads form the column. And you are adding a protein to which histidines have been added. Now the histidine forms coordinate bonds with the nickel to form this hexavalent complex, and it will get stuck. So the protein having histidine tag will get stuck to the column, while other proteins will flow out. And after you have selectively separated the protein with the histidine tag, you can add imidazole. So imidazole will replace the proteins and get stuck to this nickel, while the protein with the histidine tag will flow out. So in this affinity chromatography, you are basically modifying the protein by adding a histidine tag so that it interacts with this nickel coordinate complex compound, which is there in this column. And you are selectively separating out the histidine tag protein and eluting it later. There are several methods of protein crystallization. One is called vapor diffusion. So this is the most commonly employed method of protein crystallization. In this method, the droplets containing purified protein, buffer, and precipitant are allowed to equilibrate with a large reservoir containing similar buffers and precipitants in higher concentrations. So basically, in this vapor diffusion, it can either be hanging drop like this or sitting drop where the drop is sitting. So initially, the droplet of the protein solution contains lower precipitant and protein concentrations. But as the drop and reservoir equilibrate, the precipitant and protein concentrations increase in the drop. What this means is that, so the protein and precipitant concentration is low, and so this buffer solution that is there is evaporating from the drop and condensing in the reservoir. So this is increasing the concentration of protein in the drop, whether the drop is hanging or sitting. So if the appropriate crystallization solutions are used, with increase in concentration of the protein can form crystals in the drop itself. This method is used because it allows for gentle and gradual changes in concentration of protein and precipitant concentration, which aid in the growth of large and well-ordered crystals. So basically what this is saying is that in this method, the evaporation of the buffer solution is very slow, and this allows the crystals to form well-ordered structures. Whereas if the evaporation was fast, then the crystals would be haphazard. So as I told before, that vapor diffusion can be performed in either hanging drop or sitting drop format. The hanging drop apparatus involves a drop of protein solution placed on an inverted cover slip, which is then suspended above the reservoir. In sitting drop crystallization apparatus, the drop is there on a pedestal that is separated from the reservoir. Both these methods require sealing of the environment so that an equilibrium can develop between the drop and the reservoir. So basically, this system is completely isolated from the outside environment, and only then this equilibrium can occur. Another method is called microdialysis. So this takes advantage of a semi-permeable membrane across which small molecules and ions can pass, but proteins and large polymers cannot pass. So a gradient of solute concentration is established across the membrane. So what this means is this is a membrane and you have the protein concentration inside, and which is very low, and the protein concentration outside is high. So what will happen is that the buffer solution and small ions will pass outside in order to equilibrate this concentration. And this will result in increasing the concentration of the protein inside the membrane. In this way, this protein solution is slowly moving towards supersaturation, and that enables the protein crystals to form. So the difference between microdialysis and vapor diffusion is that in microdialysis, a semi-permeable membrane is involved. But in all three, the common factor is that there is an equilibrium forming between the protein solution and the reservoir. And due to this equilibrium, there is a supersaturation occurring in the protein solution, which leads to the formation of the crystals. Now, there are several zones of crystallization, which we will study now. The solubility line, which is the dark blue line, divides this diagram into an undersaturated, which is the white background, and supersaturated zone. So this white background is undersaturation, meaning that you can add more of the protein molecules and it will still be soluble. And the protein molecules in this undersaturated zone stay in the solution. With increasing, just 1 second, I think someone wants to join. If you can just allow them to join. 1 second. Right, so I'll continue. Yes, so as I was saying, this there is a dark blue line which defines the difference between undersaturation and supersaturation. In undersaturation, you can keep on adding protein molecules and it will still be soluble. Whereas when you go into the supersaturation, that does not happen anymore. So in supersaturated region is subdivided into a metastable state, which is shown by these two blue colors, and an unstable state shown by this red color. In this metastable region, there is a region of crystal growth, which is this light blue region. And there is a nucleation zone shown by this dark blue region. So in the crystal growth zone, only transient nuclei form that do not reach the critical size. If the supersaturation proceeds, you eventually reach the nucleation zone, where the nuclei reach a critical size and eventually leads to the formation of large crystals. So as the nuclei become larger and the crystals start to appear, the protein concentration in the solution is decreased and the system reaches the crystal growth zone again. In this zone, the crystals will continue to grow at this lower supersaturation. When the supersaturation is too high, the unstable zone is reached, where the proteins precipitate amorphously. So basically, from an undersaturated zone, where you can continue adding more proteins, you can, if you saturate it a little more, you will reach the crystal zone, crystal growth zone. But very high saturation will give you the nucleation zone, where proteins will rapidly form from a nucleus and it will then go back to the growth zone. But if the supersaturation is also reached to its limit, the proteins will precipitate out in an amorphous format and the crystal will not form. There are many common challenges in protein crystallization. Firstly, if the supersaturation is too high, then too many nuclei form and you have many tiny crystals. Basically, you need few nuclei so that you can get large crystals. But in high supersaturation, there are too many nuclei forming and you get tiny crystals. Secondly, in many supersaturated solutions, the spontaneous nucleation does not occur. So you need to add a small crystal of the protein, which is called a seed, so that the crystal can grow around that seed. So these are some challenges faced in the protein crystallization. Now, you know about the unit cell in crystal, which is the smallest repeating portion of a crystal lattice, which is the basic building block of a crystal. So if this is your crystal lattice, this shaded portion is the unit cell. The unit cell is characterized by the axes, which is A, B, and C corresponding to the X, Y, Z axis, and the angles between the axes shown by alpha, beta, and gamma. So by defining these 6 parameters, A, B, C, and alpha, beta, gamma, you can get an idea about the dimensions of the crystal lattice. And the whole crystal can be generated by unit translation of this unit cell in the 3 dimensions. Now, now there is something called a Structural Genomics Research Consortium, or SGRC. This was an initiative taken by the Protein Structure Initiative of NIH USA to determine the 3-dimensional structures of medicinally, health-related, and biotechnologically important proteins. This is basically like the Human Genome Project, but while the Human Genome Project was to determine the structures and conformations of the human genetic makeup, this is to find that for the biologically important proteins. This aims to place all its research output into the public domain without restriction and does not file for patents and continues to promote open science. So this is a publicly available platform and there are no restrictions to it. There are several goals of this SGRC. Firstly, it aims to classify all available genomic sequences to establish a prioritized target set. So basically, only when you know all the genomic sequences, you can find out that which genomic sequence is causing the disease. So then you can target the disease by targeting those specific genomic sequences. The second thing, it aims to clone and express the genes and gene fragments of microbial and eukaryotic origin. So you learned about cloning through plasmids of E. coli, and that is done to express many different copies of the DNA for study. Then it aims to purify and crystallize native and derivatized proteins for X-ray crystallography. So in order to understand the structure of the protein by X-ray crystallography, you need the crystals. Then it collects data and determines the protein structure. And finally, it analyzes the protein structures to understand the fold and function and also predict how the protein can evolve to further modify its function. So these are the goals of the SGRC. Now in crystals, so as you learned a few slides ago, that diffraction from a single molecule gives very low intensity patterns, and so it's not enough, and you need diffraction from a crystal. So how does diffraction from crystal increase the intensity of the final diffraction pattern? So there is something called Bragg's law. So if you look at these two light rays, one of the, or if you consider them X-rays in this case, this X-ray is reflected from the upper surface of the crystal and it reaches your eye. But this X-ray comes and is reflected from the lower surface. So this X-ray travels an extra distance, which is equal to 2d sine theta. This extra distance of 2d sine theta that is traversed by this X-ray can be of different wavelengths. If the wavelength is an integral multiple of the wavelength of the X-ray, then there is constructive interference. What constructive interference basically means is that you have two X-rays and their peaks and their troughs are corresponding. And so when you get your final diffraction pattern, the amplitude will be twice. Because the amplitude of the two X-rays are at the same place and this lower trough is also at the same place, your net amplitude will be twice that of either of these X-rays. So this is called constructive interference, and it can only happen when this extra 2d sine theta distance is an integral multiple of the wavelength of the X-ray. So in case of constructive interference, your intensity increases, shown by the increase in amplitude, and that is why from a crystal you will get many such X-rays which have constructive interference, and the overall intensity of your X-ray diffraction pattern will increase. So in the crystal, the ordered periodic arrangement of molecules produces constructive interference, and because of the constructive interference, you will get a strong diffraction pattern. The interference is constructive because the path length difference, which is 2d sine theta, is an integral multiple of the wavelength n lambda. This situation is only possible because the diffracting objects are periodic. Now, if there is an irregularity in the crystal, which can be of different types, like it can be rotational disorder. Like if you look at this diffraction pattern, in rotational disorder, the diffraction peaks are shifted rotationally, and you can see there is a blurring around the rotational direction. In translational disorder, the diffraction peaks are shifted translationally, and so there is a blurring in the translational direction. So these irregularity caused by the rotational or translational disorder limits the usefulness of a crystal, because the disorder destroys the periodicity and you get diffraction patterns like this, which are weak and blurry, and they are not of much use. So the more perfect your crystal is, the better diffraction pattern you will get. So what makes crystallization such a difficult challenge? So this is a basic schematic diagram showing proteins in solution and proteins in crystals, which are in an ordered periodic arrangement. If you look at the thermodynamics of this crystallization process, delta G is delta H minus T delta S. Now delta H is basically the enthalpy change of formation of the crystal, and this is favorable from 0 to around minus 17 kilocalorie per mole, because as the crystal is forming, there are many intermolecular interactions, which can be hydrogen bonding, van der Waals, ionic, etc., which is stabilizing the crystal. And that is why this enthalpy of the crystal formation is modestly favorable. Now, entropy is of two types. One is the entropy change in the protein. The entropy change in the protein is strongly unfavorable, from plus 7 to plus 25 kilocalorie per mole, because in the solution, the protein is free. It has 3 degrees of freedom in X, Y, and Z axis, and it has a lot of entropy. But when it crystallizes, the degree of freedom in both orientation and translation becomes 0. That is because the protein is fixed at the crystal lattice points, and so the entropy considerably decreases. So the entropy change in the protein, delta S protein, is strongly unfavorable. On the other hand, the delta S of the solvent is strongly favorable, from minus 7.5 to minus 50 kilocalorie per mole. This can be understood by this diagram. So if you consider this as two proteins which are in solution, The solvents are trapped between the proteins, forming hydrogen bonds. But when the proteins come together in a crystal, the solvents are free. So here, the degree of freedom of the solvent is 0 because it is fixed in position by hydrogen bonds. But here the solvents are free and they have 3 degrees of freedom in both orientation and translation. So the entropy change of the solvent is favorable and the entropy increases. So basically, if you look at this thermodynamic equation, the enthalpic change of the crystal formation is small, but it is favorable. So it is shown by negative. While the delta S protein term is large and it is unfavorable. So it is shown by a positive term. The delta S of the solvent is also large and it is favorable, shown by negative. So if the two large terms cancel out, you can see that the free energy of formation of the crystal is small, but it is modestly favorable. Because of this, crystallization is a pretty difficult challenge. So how can you improve the chance of getting these crystals? So one of the methods is to lessen the entropic penalty of this T delta S. So as you learned in the previous slide, this T delta S is unfavorable because the protein goes from having a lot of entropic freedom to having very less entropy. So how can you lessen this? Firstly, if the protein has floppy mobile termini, like if it has a lot of histidine tags, you can remove the tag, and then what will happen is that your initial entropy is not that much. So when it becomes the crystal, the entropy change is not that much unfavorable. Another thing you can do is you can take individual domains separately and crystallize them separately. So if you remember hemoglobin, it had 4 domains, like alpha and beta domains, which was like 2 alpha and 2 beta domains came together to form hemoglobin. So you can separately crystallize the alpha domains and the beta domains and then put them together to form the net quaternary structure of the protein. That also helps to lessen the entropic penalty. Another thing that you can do is that you can bridge the 2 domains and lock them together. So if the 2 domains are locked together, then they will not have that much entropic freedom in solution. And so the entropy loss on formation of the protein is not that high. And the fourth strategy is that there are several high entropy residues like glutamic acid or lysine. Basically, any amino acid which has a long side chain has high entropy because the long side chain can move in many different directions. So if you mutate those high entropy amino acids to shorter amino acids like alanine, cysteine, or serine, the initial entropy will be lesser. And so on crystallization, the entropy loss will also be lesser. And this will push the equilibrium towards formation of the crystal. There are other ways of improving the chances of getting protein crystals. So if you see this diagram, you're going from many molecules of the protein to a single protein crystal. It is represented by n molecules of the protein, which we are calling M here, to a single crystal, which has n molecules. So the thermodynamic expression for this is delta G equals delta G0 plus RT ln concentration of the crystal by concentration of the protein to the power n. So if by chance you increase the monomer concentration, that is, you increase the concentration of the free protein in solution, that will push your equilibrium towards formation of the crystal. So that is one way of improving, so increasing protein concentration to favor crystallization is one way to improve the chance of getting protein crystals. In order for the protein to be in the nucleation zone and form crystals, you need to have a supersaturated solution. And there are 3 ways to achieve the supersaturated solution. Firstly, you maximize the concentration of the purified protein. One of the ways of doing this is called Centricon centrifugal force. So you add your dilute protein solution to a centrifuge tube and invert the tube and rotate it rapidly. So the water is removed and you can see that the protein solution, which had this much volume, is reduced to a very low volume. That is, the protein solution is highly concentrated, and you can now take this supersaturated protein solution and it will automatically form crystals. Another method is called vacuum dialysis. So basically, you put your protein solution in this conical flask, and the diluted solution is put in the conical flask and it is attached to vacuum. So by vacuuming, you are removing the buffer solution that is there, and the protein is getting more concentrated. So this is vacuum dialysis. Another method is called the Amicon pressure. This is similar to the vacuum dialysis, but instead of vacuum, you are applying pressure. So you have the dilute protein solution here, and you are applying pressure through this tube at the top of this solution. So what this pressure does is that the filtrate or the buffer solution will come out from this tube and get collected in this bucket. So as more and more of the buffer solution comes out, the protein solution gets more concentrated till the point of supersaturation. And another method is called dialysis against high molecular weight polyethylene glycol. So basically, say these molecules, these blue molecules that are shown here are your impurities or solvent molecules. And on the outside, you have polyethylene glycol solution. So in order to dilute the polyethylene glycol solution, the solvent molecules will come out from the dialysis bag, and this will result in a concentration of the protein solution inside the dialysis bag. So using these four methods, you can maximize the concentration of the purified protein to achieve supersaturation. The next method is to add, the next step is to add a precipitating agent. So here the example has been shown with polyethylene glycol, which is this molecule, which is basically an ethylene glycol, which is CH2CH2OH, and many of these have polymerized together to form polyethylene glycol. Now the polyethylene glycol, if taken of appropriate size, will stabilize the protein by forming different van der Waals and ionic interactions to the protein, and this will help the protein to crystallize out. Since the protein has several polar groups exposed, the polyethylene glycol will form hydrogen bonds with these polar groups, and so it will stabilize the protein and enable it to crystallize out. So that is the role of a precipitating agent. Other than different lengths of polyethylene glycol, high salt concentrations can also be used, like ammonium sulfate or sodium hydrogen phosphate, or small organic molecules like ethanol or methyl pentanediol or MPT. The role of all this is basically to stabilize the protein molecule and enable it to crystallize out. For different sized proteins, different lengths of polyethylene glycol are used. The third step is to allow vapor diffusion to dehydrate the protein solution. So you have learned about vapor diffusion a few slides ago. Here, the hanging drop method is shown. And say the ammonium sulfate is a salt, which is 2 molar concentration in the reservoir, but only 1 molar concentration in this drop. So the water from the drop will evaporate and condense in the solution to equalize the concentration of ammonium sulfate in the drop and the reservoir. So water is basically migrating from the drop to the reservoir, and this results in concentrating the protein concentration in the drop. And like this, you are supersaturating the protein concentration in the drop and enabling the formation of crystals. Dialysis is a method, and we will learn about the principle of dialysis. So diffusion is the random thermal movement in solution called Brownian movement that leads to the net flow of molecules from higher concentrated area to lower ones until the equilibrium is reached. One very practical example of diffusion is that if you spray a perfume bottle at one end of the room, then eventually the perfume molecules will diffuse throughout the room, and the smell of the perfume can be felt from the opposite side of the room as well. In dialysis, there is a semi-permeable membrane involved, and the characteristic of this membrane is that only small molecules can move through the membrane. At equilibrium, the concentration of the small molecules inside and outside the membrane will be same, while the concentration of macromolecules, the number of macromolecules inside the membrane are constant because macromolecules cannot pass through the membrane. So this is an example of dialysis shown here, where these large molecules are the proteins. These blue balls are the solvent, and this red molecule is the dialysate. So at equilibrium concentration, the solvent comes out from the semi-permeable membrane, leading to concentration of the protein inside the semi-permeable membrane. There are instruments called dialysis cassettes, which is basically, it is this cassette shaped thing, and you can pour the protein solution inside and allow it to equilibrate in a beaker of your dialysate. So these cassettes facilitate the rapid and effective dialysis of sample volumes from 100 microliter to 30 ml. This cassette design maximizes the surface area to sample volume ratio, and you can concentrate the protein very rapidly. There are certain other methods to enable crystal growth, one of which is the free interface diffusion. So in this technique, the protein and the precipitation solutions are brought together from different ends of the tube. They are injected through either end of the channel, and equilibrium is allowed through diffusion. The two solutions come into contact in a reagent chamber, both at their maximum concentration, and this initiates spontaneous nucleation. As the system comes into equilibrium, the level of supersaturation decreases and crystals pop out like this. Another method is the microbatch crystallization. So what this involves is a very small volume of protein droplets are immersed in oil as less as 1 microliter. The reason that oil is used is because in such low volume of protein solution, evaporation can occur. And if rapid evaporation occurs, crystallization will not occur. That is why the oil is used, so that this evaporation is inhibited. The most common oils used are paraffin oils and silicon oils. The advantage of this method is that very small amount of sample is enough. And also, since in this process, in whole time, the protein is not exposed to air. It is directly just immersed in the oil. And so the protein is protected from airborne contamination. So microbatch crystallization is a method of crystallization, and it is generally used when you have very small amounts of the protein. Next, we come to microfluidics. So microfluidics is a sub-branch of fluid mechanics that studies the physics of fluid flow at the micrometer scale. Complex fluid equations reduce to simpler form because we need to neglect the effect of turbulence and gravity at the micrometer scale. There is something called droplet-based microfluidics. What happens is that when we need to access a series of reagent concentrations very rapidly so that kinetic measurements can be done at these concentrations without labor-intensive dilutions and using minimum sample, then the droplet-based microfluidics is used. Because if you just create solutions of different concentrations, firstly, you will need a lot of the sample and you will need a lot of time. So to minimize that, what is done is that there are two streams. Say you have two different reagents shown by this red and the green. So the red and the green reagents are mixed and they are separated by the dilution buffer. So there are three streams. The buffer stream prevents the reagents from coming into contact before the formation of the drops. And by changing the amount of buffer in the buffer stream, you can modulate the concentration of the drops. So like this, you can have different drops of different concentrations. So say in the first drop, you have higher concentration of the red reagent. Then you reduce the rate of the flow of the red reagent and you can get a drop with lower concentration of the red reagent. So like this, you can control the concentration inside the drop by varying the flow rates of the three streams of the stock solution. So simply by varying the flow rates, you can get different drops having different concentrations of the reagents. So this is a way to get a variety of reagent concentrations very quickly and using minimum sample so that a lot of kinetic experiments can be done. Now, it was needed to validate that this method of dilution is correct. So fluorescein dye was used as a quantitative fluorescent marker and a plot was drawn for the concentration measured and the concentration predicted. As you can see, the measured concentration and the predicted concentration are very similar. And so this is a valid method for rapidly getting a lot of reagent concentrations in a very short time and with minimum sample. And this method is called droplet-based microfluidics. Now you learned about the different zones and like you learned that how from under saturation you can go through the crystal growth zone to nucleation, and from nucleation the crystals grow rapidly. Now if the crystallization is occurring in this light blue region or the crystal growth zone, the crystallization is occurring without the formation of a nucleus. So the growth is slow, and because of the slow growth, a very ordered crystal is formed. However, if the crystallization is occurring in this dark blue region, which is the nucleation zone, the crystallization is very fast because from a nucleus, the crystals automatically propagate very fast. Due to the fast growth of the crystal, very disordered crystals are formed. So if you look at the time, this blue line is the one with nucleation. And so, as you can see, the crystals grow fast. And as discussed, because of this fast growth, the crystals are disordered. But this green line, which is the one without nucleation, shows very slow uniform growth of crystals, leading to an ordered crystals. So how can you use microfluidics to observe the effect of mixing on the nucleation of protein crystals? Firstly, like you have learned that you can have three streams having the protein, salt, and the buffer, and you can get drops of varying concentrations. Then what is done is a cycle of advection, which basically allows the protein, buffer, and salt to mix within the drop. And the total number of nucleation events depends on the area and lifetime of the interfaces. So it is shown here that a plug-based microfluidic system is suitable for observing mixing effects in crystal nucleation. So if you set up a system like this, where you have 3 streams carrying the protein, salt, and buffer, you can set up hundreds of crystallization experiments in a short period of time, because each drop is a crystallization experiment. And it's very, it is not at all labor inducive, and you can use as less as 1 microliter sample of the protein solutions. So like this, you can set up hundreds of crystallization experiments at the same time. What was observed from setting up so many crystallization experiments is that nucleation is very sensitive to flow rate and mixing. When the flow was low, nucleation occurred rapidly and the precipitate was visible. So during slow flow, many nucleation events occurred, and so there were many tiny crystals, as you can see from this example. So in this example, the flow of the drop was slow, and this led to the formation of many nucleus, and from many nucleus, many tiny crystals were formed. But if the flow of the drop is fast, as shown here, the number of nucleation events is very few. And so from those few nucleation events, large crystals can grow after 8 hours of incubation. So depending on what kind of crystals you need, if you need many tiny crystals, you need to keep the flow slow. And if you need a few large crystals, the flow needs to be fast. So in this way, you can control the nucleation events by the rate of the flow in the microfluidic tube. Several microfluid tools have been developed to screen the protein crystallization conditions. First thing that was done is the precipitant concentration was varied. So a microfluidic system was set up in which polyethylene glycol, buffer, the protein lysozyme, and salt NaCl was added. So you can control the flow rate of any of these 4 things, and you can get drops with different concentrations. So the precipitant here is polyethylene glycol. So suppose in the different drops, you have different concentrations of polyethylene glycol. And if you see the new crystal formation in each of the drops, you can find out that what is the optimum concentration of polyethylene glycol for the best crystallization. In the example shown here, this concentration of polyethylene glycol seems to be the best because the best crystals are formed here. So in a single microfluidic experiments, you can test many different concentrations of polyethylene glycol. And this is called a gradient screen of crystallization conditions. Another method is called a sparse matrix screen in nanoliter plugs. So basically, suppose you don't know the kind of precipitant or salt that you need for crystallization. In a capillary, you can have different drops of different precipitants, each around 10 nanoliter volume. So you can have these different drops passing through the tube, and you add your protein to each drop, and you see where the crystal is forming the best. So in this example, this orange precipitant or salt seems to be the best because the crystal is forming here. So using this preformed cartridge, you can test the conditions of many different kinds of precipitants or salts and find out which is the best for crystallization in a single microfluidic experiment. Now both these can be combined into a hybrid method, and this combines the sparse matrix and the gradient screening. So what this does is that there are large drops of a certain kind of reagent, and they are separated by these spacer plugs. So you can have a certain kind of reagent, and you vary the concentration of the reagent to optimize the crystallization concentration of this reagent. Then after a spacer plug, you can have a second kind of reagent and repeat the procedure and find the optimum crystallization condition in the second reagent. So like this, in microfluidics, several different conditions can be optimized using the hybrid method. And you can find the best conditions to obtain the best crystals. Now, there are certain very distinctive differences between the crystals of salt and protein. Firstly, there are certain dyes which are available and stain only protein crystals. One of them is the ISIT dye. So this left diagram shows protein crystals before dyeing, and this right diagram shows after adding the ISIT dye. So ISIT dye selectively stains protein crystals, and this shows that this is the crystal of a protein. Another method is called crushing. So since you know that in protein crystals, 70 to 80% volume is basically solvent, so you can use a fine needle, such as an acupuncture needle, and crush the crystal. The salt crystal will crush with very much difficulty and into few pieces, while the protein crystal will be completely obliterated and have a shower of very fine pieces. So here you can see a protein crystal, which after crushing just basically becomes like a powder. Similarly, if you allow a protein crystal to dehydrate in air, it will again completely become like a powder, but the salt crystal will dehydrate intact. This is again because the protein crystals have almost 70 to 80% solvent. The ultimate test of finding whether the crystal is salt or protein is the X-ray beam. So the periodicity of protein crystals are very different from that of salt crystals. And here you can see an X-ray diffraction pattern of a protein and that of a salt crystal. So clearly the electron densities are much higher in the protein crystals. Now that you have learned how to optimize crystallization conditions and get the best crystals, you need to learn about mounting crystals. So basically a loop is used to pick up the crystal from the buffer solution, and then the loop is after you have the crystal picked up in the loop is removed by a small hook or forceps. This is called mounting crystals. And so this loop is very necessary because without the loop, unwanted dehydration of the crystal can occur. Other than loop, there are different ways to mount crystals. This is called a glass fiber, or a two-stage fiber, or a fiber topped with lengths of glass wool. It can even be mounted within a capillary tube. But the most common method is called this solvent loop. So the most modern instrument is called an adjustable mounted cryo loop with easy snap microtube. So this allows you to manipulate and orient this loop in multiple unique positions, which was previously unattainable with typical cryo setups. The loop is 20 micron in diameter inside a malleable stainless steel sleeve, and there is an easy snap microtube. This microtube is 24 millimeter in length, and you can adjust it to different millimeters, like you can extend it or shorten it to best help you to pick up the crystal. So in this way, using this instrument, you can optimize the process of mounting the crystals on the loop. The next step is cryopreserving the protein crystal. So why do you need to freeze the protein crystal? Basically, X-rays have very high energy and they can damage the crystal. So to ensure that the crystal does not decay during data collection, this freezing is very essential. So to do that, you're mounting the crystal on this stand, and then the stand is kept in a holder, and the holder is placed in liquid nitrogen in order to bring the temperature down to minus 70 degrees Celsius. The crystals must be flash frozen. So the water must be frozen to minus 70 degrees Celsius very fast. So why is this? So 70 to 80% of the protein has solvent, which is basically water. Now, if you freeze the water slowly, hexagonal ice will form, and that will completely destroy the orientation of the water in the crystal. Contrary to this, if you suddenly freeze the water at minus 70 degrees Celsius, something called water glass forms, which is basically that the water will be retained in the same position as it is there in the crystal, and it will just suddenly be frozen in that same position. So how is this flash freezing done? The crystals which are mounted on loops are flash frozen by dipping in liquid propane or freon at minus 70 degrees Celsius, or by instant exposure to nitrogen gas at minus 70 degrees Celsius. What this will do is that it will instantly freeze the water in the same orientation as the crystal. And so the crystal orientation will be maintained. On the other hand, if the freezing was slow, then hexagonal ice would form and it would completely destroy the crystal orientation. There is an instrument called goniometer in crystallography, which is used to measure the angles between crystal faces. They are also used in X-ray diffraction to rotate the samples. Just give me 1 minute. So basically, what is done in a goniometer is that once you have a suitable crystal, it is attached to the goniometer head or the sample holder in an arbitrary orientation. The goniometer head is then placed on the base of the goniometer assembly, and the crystal is optically aligned in the center of the incident X-ray beam using a video camera or a microscope. The orthogonal XYZ translations on the goniometer head are adjusted until the specimen is centered on the crosshairs for all crystal orientations. The most common type of goniometer is shown here, which is the 3-circle goniometer. It offers two angles of rotation: the omega angle, which rotates about an axis perpendicular to the beam, and the phi angle about the loop or capillary axis. So this is your goniometer head where the crystal is mounted, and the rotation along that axis is the phi axis, while the rotation along the direction of the X-ray, perpendicular to the direction of the X-ray, is called the omega axis. The third angle is fixed as the magic angle of 54.74 with respect to this omega axis. So the third angle is measured with respect to omega axis, and you can see it is 54.74 degrees. Now that you have mounted the crystal and passed the X-ray, so you will now get a diffraction pattern. So the basic principle of X-ray crystallography is that you have the protein crystal, which is the most difficult part of the process to achieve a perfect crystal for, or as close as perfect to possible for analysis. The crystal needs to be as close to perfect because the position of the electrons must be mapped accurately. So a flawless structure is important. So on passing the X-ray through the crystal, you get a diffraction pattern like this. In this diffraction pattern, the spacing of the different spots is used to get the unit cell dimensions. Note that the diffraction pattern has a certain symmetry, and this is because of the symmetry of the protein crystal. So once you have the diffraction pattern, each of the dark spots correspond to the position of an atom, and the density of the spots varies with the amount of interference with the diffracted electrons at each point. So where there are many dark spots, the electron density is high, like it can be a phenyl ring, which has high electron density. And where the spots are sparse, the electron density is lower. So once you have the diffraction pattern, it is analyzed to get an electron density map. And finally, from the electron density map using Fourier transformation, it is transformed into a three-dimensional model of the protein. Now, the protein model can further be used to get another diffraction pattern to get a more refined protein model. And this process can be repeated several times to get as much good resolution of the protein model as possible. So now we will briefly talk about X-ray discovery and production of X-rays. So how the X-rays are produced? Basically, uh, this is called a cathode ray tube, where a voltage is applied to this filament. So this voltage is heating up this filament, and when it is hot enough, electrons are emitted. This is called thermionic emission. When the electrons are emitted, they are accelerated through a high potential difference of around 50 to 100 kilovolts towards a metal target called the anode, which is positively charged. So there is a positively charged target and the electrons are attracted towards it and accelerated. The anode is usually made of tungsten and is made positive with respect to the filament, attracting the negatively charged electrons and causing them to accelerate. The electrons then collide with the anode and rapidly lose the acceleration, and thus kinetic energy is lost. And this kinetic energy that is lost is observed in the form of X-rays. So basically, X-rays are formed by accelerating electrons hitting a metal target and losing the kinetic energy, which is released in the form of X-rays. If we look at the atomic basis of formation of the X-rays, what happens is that this electron that is accelerated uh, comes and hits the atom, and it knocks out one of the electrons from the inner shell of the atom. So this electron that is hitting the atom is basically this electron from the fragment which is accelerated. And after it knocks out one of the electrons from the inner shell of the atom, this electron is also deflected outside. So then a vacancy is created in the inner shell. In order to fill this vacancy, one of the electrons from the outer shell jumps into the inner shell. And this jumping releases an energy which is basically released as X-ray. So if an electron jumps from the second shell to the first shell, the energy difference between these two shells is released as an X-ray. If it jumps from the third shell to the first shell, the energy difference between the third and first shell is released as an X-ray. So X-rays have very characteristic definite energies. And this is the basis for the production of X-rays. So now that we have gone through the lectures that were taught in week 3, we will now deal with a few sample problems. So I'll go to the first sample question, which is static information about a protein structure can be obtained from which of these following methods? So I'll give you some time to work it out and then I'll discuss the answer. Okay, so the answer is A, which is X-ray crystallography. And So static information about the protein structure and why is it static information? So in X-ray crystallography, as you know that the method involves getting a protein crystal which is as close as perfect to possible. Then X-rays are passed through it to get a diffraction pattern. From the density of spots in the diffraction pattern, you can get an electron density map. So where the electron density is large, there will be many spots. And where the electron density is low, you will have fewer spots. So from the electron density map, you can get an idea of which groups there are. So suppose in regions of high electron density, it can be an aromatic group and so on. And from the molecular modeling, you can get an idea of the final 3D structure of the protein using Fourier transformations. So basically, since in crystal, the protein structure is fixed or static. So as you know, in solution, proteins are allowed to move about and have dynamism. But in crystals, the protein structure is static. So whatever data that you are getting from the protein crystal is a static information. And so X-ray crystallography gives you static information about the protein structure. On the other hand, NMR, circular dichroism, and UV-Vis spectroscopy are all done in solution. And in solution, the protein can move and have dynamism and rotate in different ways. And so whatever structure you get from NMR, CD, or UV-Vis spectroscopy is basically an average of all the conformations adopted by the protein. And so that is dynamic information. We go to the next question, which is asking that which of the following is true for a protein crystal? Again, I'll give you some time to work it out and then discuss it. Okay, so the correct answer is A, which is basically saying that in a protein crystal, the protein remains functional. So as we learned before, that in a protein crystal, around 70 to 80% of the crystal is basically solvent. And because of that, there are not that many intercrystal forces causing rigidity in the protein. So even in the crystal state, the protein can accommodate conformational changes, and that is what enables the protein to remain functional. So if you look at this, this is showing a target enzyme, and this crystallization was done in vivo, that is within the cell itself. And from this crystal structure, it was observed that even while it is a crystal, it can catalyze the transformation of a substrate to a product. So for this, as you know, the enzyme has to have certain conformational changes and flexibility in order to accommodate the substrate and transform it to the product. And because of the high solvent content of the protein, it can accommodate these conformational changes and remain functional even in the crystalline state. So clearly B is wrong, because protein is present in the molten globule state. Molten globule is a state during folding of proteins, when basically from an unfolded protein, when all the hydrophobic groups come together, that is called a molten globule state, and it is an intermediate in folding of protein. So that has nothing to do with protein crystal. And C is also wrong because protein is denatured only at high temperatures or if we add some denaturing agent. So crystallization is done in mild conditions, so protein is not denatured. So clearly the correct answer is A. Then we'll go to the next question, which is asking that which of the following tag do you incorporate in your protein for nickel NTA affinity chromatography? So the correct answer is C, histidine tag. So if we recap back into the method of nickel affinity chromatography, in the column, there are certain beads which have this nickel coordinate complex. And when the protein is tagged with a histidine tag, it can bind to the nickel, forming coordinate bonds. So then what will happen is that if you have a protein with the histidine tag, that will selectively get stuck in the column and the other proteins will be eluted out. Now, now you have the column with the protein histidine tag stuck to it. If you now add imidazole, the imidazole will replace the protein with the histidine tag. And finally, you can get your selected protein with the histidine tag out of the column. So basically, it is the histidine imidazole ring that is interacting with nickel to form this complex that is selectively allowing the protein with the histidine tag to be separated from the other proteins. So the correct answer is C, histidine tag. The fourth question is that which of the following amino acid side chain resembles the imidazole ring? So the correct answer is D, histidine. So if you look, this is the structure of an imidazole ring, which is a 5-membered ring with 2 nitrogens at the 1 and 3 position. The histidine side chain consists of a CH2 and an imidazole ring. The imidazole ring is what is making the histidine basic. So here you have the zwitterionic form of the histidine with the net charge 0. Now, in more basic environments, this NH3 plus loses its H and it becomes a net negative charge of -1. And this happens at pKa values around 9.17. In acidic environments, this histidine imidazole ring gets protonated and the net molecule has a negative charge of +1. And this happens at pKa values around 6. At even lower pKa values, meaning more acidic conditions, this CO- will also get protonated. And at pK, this happens at pK 1.82, and the molecule has a net charge of +2. So this is called a titration curve of histidine, where as you add more equivalents of the base, gradually the charge of the histidine moiety goes from +2 to -1. The next question is that which of the following statements is false when performing size exclusion chromatography? So the correct answer of this is B. So to recap, in size exclusion chromatography, you have these porous materials, which is acting as your filter, and the pores in these materials can accommodate small molecules, but the large molecules cannot enter the filter. So the large molecule is eluted first, that is, it comes out of the column first, and then eventually the smaller molecules come out later. So in size exclusion chromatography, smaller protein is eluted first, that is false. Once, yes. And sorry, in which of the following statements is true? This should have been true. I think there is a mistake in this. So which of the following statements is true? Bigger protein eluted first, that is true. And next, we will go to the next question, which is that which one of these is used to pick up a crystal from the crystal drop? So the correct answer is D, loop. So if you recap, so basically if you have, this is your buffer solution with the precipitant and your protein crystal, you also call it the host matrix. So you use a nylon loop to pick it up, and then you transfer the crystal to a drop of oil. So in this oil, this host matrix is removed, and you have the loop having the crystal and the oil. So then this is mounted onto the goniometer, and then you can find out the angles between the different faces of the crystals using the goniometer. So it is the loop which is used to pick up a crystal from the crystal drop. The next question asks that if we look at the thermodynamic equation of crystal growth, which is delta G of formation of the crystal is delta H, which is the enthalpy change of crystal formation, minus T delta S. And delta S is a summation of the enthalpy change of protein plus enthalpy change of solvent. So which is correct about that thermodynamics of crystal formation is, which is correct? So the correct answer is C. That is, it is modestly favorable. So if you recap, uh, this is the equilibrium of protein in solution to the crystallized protein. So this enthalpy of formation of the crystal is small, but it is favorable because in the crystal, there are several intercrystal interactions, which can be hydrogen bonds, van der Waals, or ionic interactions, which stabilize the protein crystal. And so it is favorable, shown by the negative sign. The enthalpy change of the protein is unfavorable, and it is large. It is unfavorable because the protein is going from having a large entropy, which is basically it has 3 degrees of freedom in both orientation and translation in solution, while in crystal the protein is fixed. That is, it has 0 degrees of freedom in both orientation and translation in the crystal. So the entropy change of the protein is unfavorable, and it is large. So it is a positive large value. The entropy change of the solvent is also large, but it is favorable because in solution, the solvent is solvating the protein and it is fixed to the protein by hydrogen bonds. So the degree of solvation of the solvent is 0 in solution. But in the crystals, when the proteins come together, the solvent is now freed up and it now has 3 degrees of freedom in both translation and rotation. So the entropy change of the solvent is favorable, shown by the negative sign, and it is large. So if the two large values, positive and negative, cancel out, the free energy of formation of the crystal is small, but it is favorable. So the correct answer is C, modestly favorable. We next go to question 8, which is asking that which of the following is not an enthalpic term for the enthalpy of formation of the crystal. The correct answer is B. So in formation of the crystal, uh, you can have hydrogen bonds, which will contribute to the enthalpy change of formation of the crystal. Or you can have lattice contacts, that is contact between the different lattice points. And this will also add to the enthalpy of formation of crystals. However, the degree of freedom is something that is associated with the entropy change of formation of the crystal. So the degree of freedom refers to translation or rotation along the X, Y, and Z axis. And this is associated with the entropic term, so the delta S. So clearly, the degree of freedom, or B, is not an enthalpic term for the enthalpy of formation of the crystal. The next question is that what is the value of the third angle, or the magic angle, of the goniometer? The correct answer is A, 54.74 degrees. So if you remember the different angles of the goniometer, in the most common type of goniometer, which is the three-circle goniometer, there is the omega angle, which is rotation about an axis perpendicular to the beam, the phi angle, which is rotation along the loop or capillary axis along which the crystal has been mounted. The third angle, or the magic angle, is this angle of 54.74 degrees with respect to the omega axis. And so the correct answer is A, 54.74. The next question is that in which of the zone protein crystals nucleate and grow? The correct answer is B, nucleation zone. So if we recap and remember the different zones of the protein concentration, firstly, you have the undersaturated zone, where the more protein crystals you add, the more it will go into solution. Then it enters the supersaturation zone, in which you have the crystal growth zone, where the crystal grows without the formation of a nucleus. And so the growth is slow and ordered crystals are formed. Then you have the nucleation zone, where a nucleation is formed, a nucleus is formed, and this leads to rapid growth of the crystal, or a very disordered crystal is formed. So in this nucleation zone, which is a degree of supersaturation when a nucleus is formed, this is the zone where the crystal nucleates and goes into the growth zone. And if the supersaturation increases more than that, it becomes unstable and reaches the precipitation zone, where the protein will precipitate out in an amorphous form. So protein crystals cannot grow in the precipitation zone, because in this zone, the protein precipitates out in an amorphous form. And metastasis zone is not a zone at all within these zones. So nucleation zone is the correct answer, where the protein crystals nucleate and grow. The next question is that which among the following techniques is not a high resolution structure determination technique? The correct answer is D, circular dichroism. So if you recap, the three methods for high resolution structure determination are X-ray crystallography, NMR, and cryo-electron microscopy. To just go through the differences between these methods is that in X-ray crystallography, the sample must be crystallized in a solid frozen structure. In NMR, the sample must be dissolved and radioactively labeled with NMR active nuclei. In cryo-electron microscopy, the sample is frozen in its native state. X-ray crystallography and cryo-electron microscopy can deal with any size macromolecule, but in NMR, there is a certain size limit. And that is, it can only deal with small molecules. And other than that, in X-ray crystallography, you get atomic resolution, but crystallization can, but getting an optimum condition for crystallization can take years and even damage the protein structure. In NMR, you get close to the real protein structure, but larger proteins cannot be resolved because larger proteins tumble very fast, and that leads to overlap in NMR signals. In cryo-electron microscopy, you get near atomic resolution and the sample preparation is also very fast. So it is the most modern technique. So clearly A, B, and C are all high resolution structure determination technique. So the incorrect option is D, which is circular dichroism. The next question is that according to the electromagnetic spectrum, which of the following has shorter wavelength? The correct answer is C, X-ray. So if you look at the electromagnetic spectrum, you can see that microwaves are pretty large, having 10 to the power -1 meter length. Then you will find it's visible with 5 into 10 to the power -6 meter length. Then comes the UV radiation with 10 to the power -18 meter length. And the shortest wavelength is that of the X-ray with 10 to the power -10 meter length. So the correct answer is clearly C, X-ray. The next question is that according to the electromagnetic spectrum, which of the following has more resolving power? So the correct answer is again B, X-ray. And so if you look at the different like atomic size of atomic nuclei, atoms, molecules, you can see that a microwave can only distinguish like between humans or buildings, while radio waves are even larger. And visible lights can distinguish between unicellular organisms, but it cannot identify molecules or macromolecules. It is only X-ray, which has the highest resolving power in these options, that can distinguish the different atoms and biological macromolecules. So the correct answer is B, X-ray. The next question is that which of the following statement is false? The correct answer is C. This is because if you remember towards the beginning of this session, I had discussed that a single molecule is a very weak scatterer of X-ray. And because most of the X-rays pass through the molecule without being diffracted. And that is why the diffraction pattern from a single molecule has very low intensity. And so A, B, and D are all correct. So the only incorrect option is C. And as you can see, we use crystals in X-ray crystallography because in crystals, there are many number of protein molecules. And that enables, and through constructive interference of the diffraction patterns through all the molecules, we get a much more intense pattern from the crystal compared to individual protein molecules. The next question is, which of the following is not true for X-ray crystallography? The answer here is B. As we had discussed, X-ray cannot be focused using lenses. Like in normal light microscope, the light can bend and focus at a focal point. But in X-ray, because the refractive index is near 1, the light will not bend. The X-ray will not bend. And so it cannot be focused using lenses. So B is incorrect, that X-ray can be focused using lenses. And we have the final question for today, that in the vapor diffusion method of protein crystallization, a drop is allowed to equilibrate with a large reservoir containing buffers and precipitants. Which of the following is not a constituent of this droplet? The correct answer is A, denaturating agent, because obviously in the drop you will have your protein and your buffer and your precipitant, but a denaturating agent will only denature the protein. So obviously you will not have denaturating agent in the drop. So your correct answer is A. So that brings us to the end of today's session. Please feel free to contact me with your doubts, and I hope to meet you for the next session.